Hello and welcome everyone to this podcast for genetics. In the past two podcasts, we talked about gene regulation in bacteria and more specifically, the lac operon. Now what I want to talk to you about are lac operon mutations. In the last podcast, I explained how the lac operon worked in great detail. But how did Jacob and Minode figure out the function of the lac operon? They did it by generating many different mutations. I'm not going to go into detail on how they created the mutations. I just want to talk about the different mutations they discovered and how that affected the metabolism of lactose. Okay, so as we begin, let's go ahead and draw the lac operon. And let's draw all of our favorite parts of the lac operon. We have our promoter here. We have our operator here. And then we have our three genes, Z, Y, and A. Off to the side here, let's draw our inhibitor repressor and our activator cap. Over here on the left, let's write the various mutants that we want to talk about. And then over here, we're going to talk about the phenotype. So our lac Z minus mutants and our lac Y minus mutants. Then we'll talk about a mutant called I minus, another mutant called I superscript S. Then we'll talk about a mutation called OC. The C stands for constitutive. And then we'll talk about a mutation called minus. We might talk about some more too, but this will get us started here. I'm going to draw a phenotype over here, which will give me some room over here to draw some things. Okay, our lac Z mutant, Z minus, will make no beta galactosidase. So if it's not making beta galactosidase, the cell won't metabolize lactose. Because remember, if we have our cell here and lactose outside of the cell, it'll still enter the cell because we're still having the permease being made. But once inside the cell, it can't be metabolized into our two monosaccharides of glucose and galactose. And so it won't be able to metabolize lactose. And if that's the only sugar present, that would be lethal to the individual E. coli cell. Now the Y mutant here, Y minus, that would make no permease. This turns out to be pretty bad for the cell. Because if we have our cell here, we have our lactose outside of the cell, it can't get into the cell because remember, permease, the Y gene here, usually makes a channel to let lactose in. And if there's no lactose permease present here, all the lactose stays outside of the cell. And as a result, beta galactosidase can't do its job inside of the cell. So lactose cannot be metabolized because it's stuck on the outside of the cell. So here we can say lactose can't enter the cell. I'm going to go a little out of order here. I'm going to talk about P minus now. P minus is when the promoter is mutated. So if the promoter is mutated, RNA polymerase can't bind. And as a result, if RNA polymerase, as well as cap, I might as well write that here too, cap and RNA polymerase can't bind to the promoter. If we can't bind to the promoter, then lactose genes, Z, Y, and A, are always off. Doesn't matter if there's lactose in the cell, glucose in the cell, or both in the cell. These genes will never ever be turned on because there's no promoter. Now to explain these other three, I minus, IS, and OC, I need a little bit more room to draw. So I'm going to erase these words here. And I'm going to go and get rid of Z and Y because we've already talked about them, and P. So all we have left are I minus, IS, and OC. So let's talk about these I mutations first. I minus has the phenotype. So in the I minus mutation, the lac operon is always on, meaning that the RNA polymerase can always transcribe these genes. And let me explain why that is the case. Up to now, I've always drawn, kind of in a generic way, the repressor looking like this. However, that's not very accurate. The repressor really has two specific binding sites. it has a DNA binding site and a lactose binding site. Or rather, more accurately, I should call it an allolactose binding site. So when there is no lactose in the cell, this DNA binding site can bind to the operator to prevent transcription of Z, Y, and A. However, when lactose enters the cell, it's converted into allolactose. Allolactose now binds to this allolactose binding site, causes a conformational change which blocks this DNA binding site, so it falls off. So both of these active sites have to be working properly to regulate this gene, these genes. So we might as well call this I+, the unmutated form of it. I- looks like this, if I'm going to use this as my model up here, where the DNA binding site is no longer available, so it will not be able to bind to the operator, ever. If it can't bind to the operator, it won't be able to block transcription of Z, Y, and A. Even when lactose is not in the cell, these genes will be turned on. If you put lactose in the cell, it still can bind, or allolactose can still bind to this binding site, but it really has no effect because it's never on the operator because the DNA binding site has been mutated. So again, the LAC-I mutation has the DNA binding site. Let's go and draw that here. DNA binding site is blocked. So it can't bind to the operator and it can't turn off transcription. Always on. Okay. Next, let's talk about LAC-S. LAC-S is the opposite. You can think of the S as being super. It's a super repressor. LAC-S, the LAC operon, that is the gene Z, Y, and A, are always off. If lactose is in the cell, if lactose isn't in the cell, doesn't matter. These genes are always off in the mutant LAC-S. So let's draw what the repressor is going to look like in these mutants. It's going to be just the opposite. The DNA binding site is still available, but now where this lactose binding site used to be, it is now gone. And so now this LAC-S, or the super repressor, will always be bound to the operator. It will always prevent RNA polymerase from transcribing these genes. Even when you put lactose in the cell, and I'll put L here for lactose, it can't bind here to pull off the repressor because the lactose binding site is mutated and cannot bind allolactose. Super repressor, always bound, never turns on the genes. LAC-I, never bound, always transcribes the genes. Okay, we have one left on this board to talk about, and that is OC. OC, the C stands for constitutive, which means that these genes are always on. ZYA, always on. Just like the LAC-I mutation, except for a different reason. And when I say always on, that's just another way of saying constitutive, which is what the C is. And so in this mutation, the OC mutation, the operator is effectively removed. So even though you have this amazing looking repressor, and even though both of the active sites, the DNA binding site and the allolactose binding site, are both functional, it doesn't matter, because the DNA binding site can never bind here because that operator has been removed. No operator, the repressor can't bind. If the repressor can't bind, RNA polymerase will always transcribe these genes. There are two more mutants we haven't talked about. The first is CAP and the other is adenyl cyclase. Both of these mutations have the same effect. They will weaken ZYA expression. They both weaken ZYA expression because in order to turn these genes on fully, CAP bound to the inducer, cyclic AMP, will bind to the promoter and recruit RNA polymerase. If CAP is mutated, it can't bind to the promoter. If adenyl cyclase is mutated, it won't make cyclic AMP. If it doesn't make cyclic AMP, it won't be able to bind to CAP to activate transcription of Z, Y, and A. So the net effect is reduced expression of Z, Y, and A. Okay, so that ends our discussions of bacterial gene regulations, the lac operon, and the lac mutations. As a review, I'm going to create a table here and then ask you to fill in the table. And if you can fill in the table, that should be a good test that you understand the lac operon. So over here, the question I'll ask is the lac operon on? The next question is the repressor, that is that I protein, bound to the operator? The next question is cap bound to the promoter? And you should be able to answer each of these three questions under these growth conditions. No lactose, no glucose. Plus lactose, minus glucose. And the third condition is plus lactose and plus glucose. So in each of these boxes, indicate if the lac operon is on or off, and whether or not the repressor or the cap is bound to the appropriate sites on the lac operon. Then repeat this. So do this in this way with no mutations. Then repeat this table with, say, a lac Z mutation, and then with an I minus mutation, an IS mutation, and an OC mutation. And we'll throw in the P minus mutation as well. If you can answer all that, then you understand this material pretty well. So go ahead, fill these out, and then we can talk about these in class. That's all I have to say about bacterial gene regulation. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.